Okay. And in contrast to creativity, predicting longer life, neuroticism predicts poorer health. Don't worry about all the microorganisms and going home and being obsessed with hand sanitizers. It's all good. Okay? All right, so we're going to hop on to our next phylum. All right, phylum Mollusca, the mollusks. So to give you an overview of what the mollusks are, here are some pictures of them. So this is a giant clam, just underneath that one, actually I'll show you another picture of it, these got out of order, is a gumboot chiton, or a chiton is underneath this huge giant clam. Oy. Now I've got to fix it. Maybe I can't fix it. There we go. That's the offensive one. All right. Okay. So there's a chiton. It's one kind of mollusk. How many plates has that chiton got? Is it 8? 1, 2, 3, 4, 5, 6, 7. There are 8 plates there. And what's this guy? Good, it's a cone snail. Good. You can see the shell, see the foot. Oh, he's called a regular snail, a regular land snail. Yeah, the sort of thing you might find in French restaurant. Okay. Again, you can see the foot, you can see the shell. This shell, in fact, is spiraled. This is not a Pokemon, all right? It's an actual mollusk, a real mollusk. Isn't that crazy? Look at that thing. It's beautiful. Yeah. Then we've got a big old banana slug. Anybody want to go to the University of Santa Cruz? I believe this is their college mascot, banana slug. See lots of these in the Pacific Northwest. And you can almost think about this as a snail that doesn't have a shell anymore. Okay? Some of them have internal shells. And then, of course, we've got the clams. You're all familiar with clams. And we've got octopus, the blue-ringed octopus. There's a cuttlefish. There's a squid. There's another squid, giant squid, not a photograph. Wouldn't it be awesome if that was a photograph and you took it? Right? Giant squid and sperm whale. And there's a chambered nautilus that movie was based on, at least for the spiraling of the shell. So that gives you an overview of the different mollusks, what sort of organisms we're talking about. And this is, of course, the nautilus as well. Right? Anybody get that? Who's the captain of the Nautilus? Captain Nemo. Right, and it comes from what? Classic book. Yeah. Good. Written by? Some other guy. Who wrote it?
Jules Verne.
Yeah, Jules Verne. All right. Okay. Got to be careful when you use Google to Google terms. The Nautilus, you'll come up with both. Okay. All right. So mollusks then. Phylum Mollusca. It includes the slugs and the snails and the oysters and the clams and the octopuses and the squids and things related to them and similar to them. Most mollusks are marine. But you will find some on land, terrestrial mollusks, and you'll find some in fresh water. But the majority of mollusk species are marine. Okay? So they're soft-bodied animals. They don't have bones, but they occasionally have an internal shell, and sometimes they've got an external hard shell, and sometimes no hard parts at all, really. Okay. A squid has got an internal shell, and we're going to do a squid dissection in lab, and you'll be able to see that internal shell. An octopus has no real hard parts except probably for its beak, and a snail, of course, and a clam have got hard external shells.
The internal shell on a squid being the quill.
Yeah, it's something that's called a pen. I've never heard it called a quill, but yeah. So all mollusks then have got a similar body plan. And you remember we talked about the different body plan characteristics being able to use to separate out different phyla. So as soon as we hit the annelids and all subsequent phyla are triploblastic, and they are coelomates. And the majority of the phyla now are bilaterally symmetrical. There's one good exception to that, and that's the echinoderms. So, from the mollusks onwards, or really the annelids and onwards, but let's start with the mollusks. Mollusks and onwards, I don't really want you to use body plan, at least the number of cell layers, embryonic cell layers, and symmetry, or the cavity status, to differentiate the phylum to other phyla. Does that make sense? You can use it, but it's not a good diagnostic characteristic now to separate out different phyla, because all the phyla are triploblastic coelomates with bilateral symmetry. Good? Okay. So, for the mollusks then, this is what makes a mollusk a mollusk. They've all got a similar body plan. They all have a muscular foot. Now, in the case of a snail, it's pretty obvious. In the case of a clam, it's pretty obvious. In the case of an octopus, it's a bit less obvious what the muscular foot is, but it's basically the tentacles and the tenticular part. They all have a visceral mass, and that's kind of the soft part where a lot of the organs are. And then they've got a mantle, and again, the mantle can take sort of different forms and shapes, but that is usually a sort of a covering, or it's sort of the envelope that the visceral mass is held within. And I'll show you some diagrams to sort of help illustrate that. But all mollusks then have got a muscular foot, a visceral mass, and a mantle. Now, many mollusks have got a water-filled mantle, and some of them can hold a little pocket of air in there. And some of them have a rasp-like radula. Now, I want you to think of the radula as kind of like a tongue. It's found in the mouth, and it's very raspy. It's very rough, and they use that to abrade, to scrape off food that they feed on and swallow. So, here's our sort of generic diagram of a mollusk then. All right? So, here's the foot. In the case of this guy, there's the foot. It's pretty obvious. It's what it glides around on. The visceral mass is all of the soft parts, a lot of the organ systems. And there we've got a mantle, which is this layer which sort of encloses the visceral mass, but not really the foot. Okay? Now, in the case of this mollusk, the mantle secretes this hard shell. So if it has a shell, it's usually secreted by the mantle. Right? This one happens to have a radula in the mouth region, this sort of very rasp-like, tongue-like structure. Okay? And this breaks off or abrades off food particles that's pushed back into the mouth and it's swallowed, goes into the stomach, which is part of the visceral mass, goes through the intestines, and there's the anus. Gills are usually part of the visceral mass. There's a heart, usually. They've got gonads, of course, for reproduction, and there's a coelomic cavity, either the whole cavity or a small part of it inside the visceral mass. Good? Get a good picture, generalized view of a mollusk. So mollusks then usually have separate sexes. You've got male individuals and female individuals, and their gonads are in the visceral mass together with most of our other organ systems. But some snails are hermaphroditic, which means in the individual there are both male and female gonads. So we talked a little bit last time, didn't we, about the advantages and disadvantages of being a hermaphrodite. Yeah? And that still applies here. So many mollusks have a larval stage. So of course the sperm will fertilise an egg, the egg will hatch, and what will hatch from the egg is not a mini version of the adult, but a larval stage. And that larval stage is often ciliated. It's got cilia, and what do the cilia enable that larva to do? Swim in order to disperse. Yeah. So ciliated larvae enable it to disperse. The ciliated larval stage is often called a trochophore larvae. Okay. Now we've got about 93,000 known species of mollusks. And it's the second most diverse phylum of animals. What do we consider to be the most diverse phylum of animals and definitely has the most species? Arthropods, right? Which includes the insects and the spiders and the crustaceans and so on. All right, second only to them. And there are 4 major classes of mollusks, and I'm only going to talk about in any kind of detail 3 of them. All right? So the first class are the polyplacophorans, the chitons. Doesn't that just roll off the tongue nicely, polyplacophora? Yeah? You can almost call your dog or bird that name, right? I don't know. No? Not really? Maybe. Is it just me? All right. Polyplacophora. They're the chitons. Okay? The ones with those armoured plates on top. So we're not going to talk about the chitons very much. But we are going to talk about the gastropods quite a lot. That's the slugs and the snails. What's a good way to remember that slugs and snails are the gastropods? Just say it with a French accent. Because they are delicious, a gastronomic delight, right? And then we'll talk about the bivalves. Bi means 2, and valve refers to the shell. That's an easy one to remember, isn't it? The bivalves, they're like the clams and the oysters and the mussels. And then we've got the cephalopods. Now think of the cephalopods as sort of an octopus, a squid, a cuttlefish. Cephalo refers to what? Head. So all of those have what is a good recognizable head, often with really well-developed eyes. Okay, so cephalopod. What does the pod part mean?
Foot.
Foot. Yeah, good. Head, foot. Cephalopod. Okay. Good, you get an idea of the variety, and we're going to go over each class, look at their biology. So first we'll start with the chitons then. So you've got 8 dorsal plates which protect the animal. Directly underneath those dorsal plates we would find what? That secretes them. The mantle. And then right at the very bottom, the part that the animal moves on or it uses to anchor itself to a substrate, what would that part be? Foot. And between them is between the mantle and the foot you would find the visceral mass, right. That's it. Okay. So their foot is a lot like a suction cup. And underneath there's a little mouth. And they're able to use their radula to scrape algae off of rocks. And then they'll swallow it. So that's what a chiton looks like. Now you can get some really big ones and some really small ones. I remember being, I think it was in Cancun. And we're just, rummaging around on the rocks there, and we found loads of tiny little chitons, about that big. And then you go to the coast in the Pacific Northwest off of Oregon, and you'll find something called a gumboot chiton. And it's called a gumboot chiton because it literally looks like the bottom of a rubber boot. It's huge, great big thing like this. And when you flip it over, that's what you see. So here we've got the foot. There we've got the rim of the mantle. There we've got the mouth. What do you think this is? Not cilia. Cilia are microscopic little hairs. What do you think these are? Sorry?
Gills.
Good. They're gills. All right, for gas exchange. So, what's your clue that this is a gas exchange surface?
Folded.
Good. Folded. Huge surface area. And respiratory surfaces, gas exchange surfaces, are also thin and moist. Okay, that's all I'm going to say about the chitons. So now we're going to hop on to the gastropods. So about 3/4 of all living species of mollusks are gastropods. So the majority of mollusk species are gastropods. And the gastropods are the
Snails.
slugs and snails. Let's add in puppy dogs' tails, and that's what little boys are made of, right? So most of them are marine, but there are some that are freshwater and terrestrial. You're probably most familiar with the terrestrial ones because you encounter them most often. But most of them are marine, lots of freshwater ones. Now, the most characteristic feature of the gastropods is that they have something called torsion. Now, don't confuse torsion with the spiraling or coiling of the shell. Here's what torsion does. It causes the animals, usually you've got at the anterior end a mouth and at the posterior end an anus somewhere. Torsion has sort of caused the animal to fold or sometimes coil almost back on itself, so the anal opening and the mouth are on the same end, very close to each other. Okay? And then superimposed upon that, you can have a spiraled shell. And the spiraling on the shell, sometimes it goes clockwise, sometimes it goes anti-clockwise. And most of them have only one shell. And if they've got a shell, it's often spiraled. So that's nice. That distinguishes them very clearly from the bivalves, which have two parts to their shell. Some gastropods, of course, don't have a shell at all. It's either missing or it's reduced. So this diagram then shows the torsion. There's the mouth, there's the gut, and it sort of loops and folds back on itself. And so the anus and the mouth are not too far from each other, but on the same end of the organism. And then superimposed upon that, we've got the spiraling of the shell. Okay? All right, anybody know what this guy is? Not a clam. Clam would have two parts, two shells. It's a limpet. Okay, you find these in the intertidal. So it's another gastropod. Okay, single shell, but this shell is not spiraled, is it? But there's still torsion. If you were to pry that off the rock, that's what you would see. There's its foot, there's the edge of the mantle, there's the mouth, mantle cavity's up in there, and it's got tentacles that are sensory organs. Okay? Foot is a suction device. That's how it sticks on, and it very slowly moves and glides as it scrapes algae off the rock and feeds on the algae. Here we've got some abalone, okay, in a little gap in a rock. Anybody read Island of the Blue Dolphins? No?
Choice.
Yeah, I was going to say, isn't it compulsory reading for every 4th or 5th grader? No?
Only some of them.
Sorry, only some of them. It's an awesome book. I read it to my kids, and I loved it. I think I loved it more than they did. I used to force them to sit down. I used to read it. But it was a brilliant book. If you've not read it, read it. It's such an easy read. It's a 5th grade reading book, but it's a great, great story. And there's a lot of truth in it. It's based partly on a true story. But anyway, they lived on this island on the coast, off the coast of California, and they used abalone as one of their main food sources. Okay, they would dry them, they would salt them, but look at the size of that one. Huge, eh? Tasty. All right. So I want to show you how the radula works then. Now, if you have a look at our saltwater aquarium, as the snails glide across the glass, you can see the radula at work. I want to show you this video of how it works. See, it comes out as like a scraping motion. And it's like a rasp. If you do any woodworking, you'll know what a rasp looks like. But I'll show you some close-up pictures of the radula. And this one's obviously just scraping algae off the glass aquarium walls. But that radula can be quite destructive. They can mow down plants, can cause a lot of damage on wood. Okay, and there's a cartoon drawing of the radula and the muscles that are attached to it. It's quite a complex structure in terms of the musculature. So this is a scanning electron micrograph of the radula, and you can see it's these like sharp toothed plates, and they're replaced. What do you think they're made of? Probably not chitin. There might be some chitin in them. What are they most likely made of, though? Maybe the same stuff as the shell, right? And the shell of many of these gastropods has a protein component and usually a calcium carbonate component. So they're quite cheap to make in terms of the resources. Calcium carbonate's readily abundant, and obviously as they get worn away, you can just simply grow more from the base. There's another one. Looks pretty gruesome, doesn't it? Yeah. So you can see now how effective they would be at like shredding algae or even quite tough plant material, just shredding it. So some snails are quite venomous. We've got cone snails, and cone snails have quite an interesting venom. So again, we'll see if we can locate this BBC movie. They change them all the time. If it comes up, that'd be awesome. If not, we'll move on. Access denied.
Works faster than the nervous signals that carry the pain.
Yeah, it's really quick. So Nat Geos, they don't usually change them, but you've got to sit through their commercials.
Every day, women around the world spend millions of hours just collecting the water they need for their families. Each limited edition Stella Artois chalice helps provide 5 years of clean water for someone in the developing world.
Crawling along the ocean floor is a cone snail. It looks harmless, but appearances can be deceiving. 4 tubes jut out from its shell. The long one at the top is a siphon, which it uses to inhale water and detect prey. The smaller ones on either side are its eye stalks, and in the middle, a deadly proboscis. Concealed within the tip is a lethal weapon. A harpoon loaded with a neurotoxic mix so complex there is no antivenom to thwart it. It's armed with at least 100 different toxins, more than any other creature. When some cone snails go hunting, they like to burrow into the sand and wait. When a meal happens by, the cone snail fires its harpoon. The poisons quickly paralyze the fish. Once its prey is helpless, the cone snail retracts it through its proboscis until it engulfs the fish, swallowing it whole. snail is no longer hungry, and its appearance no longer harmless.
That's pretty cool, isn't it? Why do you think the venom is so fast acting?
It's so slow.
The snail's slow, it is.
Every day, women around the world spend millions of hours just
So why
Every
fast
day,
acting?
Millions of women around the world spend hours.
So the prey doesn't get away, right, snail's slow. But think about a rattlesnake. When a rattlesnake strikes and hits a small mammal, yeah, it doesn't strike, envenomate, and then hold on. It strikes, envenomates, and lets go, and the mammal runs away. Why can't the cone snail let the fish run away?
Make it a long distance.
Might swim a long distance, maybe it's very hard to track down the fish afterwards, right? In the case of rattlesnakes and small rodents, they've got a very good sense of smell. The rodents are on the ground, and the rattlesnake just uses its sense of smell to go find that rodent. Why doesn't the rattlesnake hold on?
You get hurt.
Yeah, it doesn't want to get hurt. Small rodents have got pretty big teeth and they'll fight back, they'll bite back. So in this case, the cone snail, it's quite, even though it's in a shell, there's some soft parts that are exposed, but it needs to very quickly immobilize its prey so it doesn't get away because it can't track it. Imagine trying to track something in water, right? Even if it could go after it. So it acts very quickly. That way, it doesn't get away and it can eat it right there. And there's another video you can watch as well. It's another cone snail eating another fish, but it's kind of cool. So the venom, then, is a cocktail of peptides. Now, you remember what peptides are, don't you? Yeah, they're polymers of amino acids. They're very complex molecules, and it's a whole bunch of these different peptides. Now, why it's got a cocktail, I don't know. You can speculate to the reasons. Maybe some of these polypeptides are specific to one kind of fish or different kinds of prey, or maybe it needs a whole bunch for it to be so fast-acting. But there's a whole cocktail. And what we've discovered is that some of these peptides have pain-killing properties. I guess that's kind of nice, isn't it? Before it engulfs the prey whole, it deadens the pain, makes it numb, doesn't feel anything. And I doubt the fish is actually dead when it gets swallowed. It's probably just completely immobilized. But it doesn't feel any pain. So here we got just a picture of a land snail, and this is a picture of a sea slug, a nudibranch. Now, what does this remind you of? Another organism we've seen? Yeah, a marine flatworm, right? It does. They're very reminiscent. But the marine flatworms generally are dorsiventrally flattened completely. This one isn't, even though they resemble each other. All right? Super colourful, the nudibranchs. They're really pretty. Yeah.
So for the cone snail, that came out of their eye stalks?
No, it didn't. So they've got separate eye stalks. One of those tubes was a siphon. So I'll show you this with the bivalves, but they have an in-current and ex-current siphon. It's a way for them to bring in water and then eject water after it's passed over the gills, for example. So they have a proboscis. That's what that was. Okay. All right. So bivalves then are almost exclusively aquatic. You don't really find them in terrestrial environments. You find them in the water. And they're the clams and the oysters and the mussels and the scallops. And their diagnostic characteristic is that they've got a shell that's divided into two halves, or two valves. Those valves are held together by a hinge. And there is one or more adductor muscles. And the adductor muscles are what holds the valves shut. Now, some of these bivalves have got,
Aren't they abductor muscles?
adductor muscles. They're called adductor muscles. There's no B in it. So some, like scallops, they're not really eyes, but they're little sensory organs that are able to detect difference in light intensity. So if you're a scallop, you can maybe see a shadow pass over you, and you can respond to that. It's funny, isn't it, to think of a clam as being able to respond to differences in light intensity. in effect, have some sort of vision. They don't generate images. They don't have a brain that they can see an image. So the bivalves then don't have a radula. All right, so now that's a clue as to how they might feed. So they definitely don't scrape anything off of a surface, and they don't have a distinct head, but they do have a mouth. Okay, how do you think bivalves feed then?
Spongy.
Was it?
Filter feed.
They're filter feeding, same way sponges do. Yep, filter feeders. Okay, so anything that's suspended in the water, they're able to filter it out in mucus and then move that mucus with the trapped particles to the mouth. So if we could get inside a clam, this is what we might see. So there's one of the valves, one of the shells, there's a hinge area, there's one adductor, and in this case a second adductor muscle, which contract to hold the shells closed. When you eat scallops, anybody eaten scallops? And they're kind of like this squat round structure, isn't it? What you're eating is the adductor muscle. Scallops have great big thick adductor muscles, and they chop them off at each shell, and that's what you're eating. All right, so there's the rim of the mantle. The mantle is what secretes the shell. And here we've got the gills. And in this case, the gills serve sort of a dual function. So let's have a look at their biology. Where's the siphons? So there's an in-current siphon and there's an ex-current siphon. So water is drawn in, circulates around the visceral mass, but they want to circulate it over the gills. That way you're going to get gas exchange. And then the gills are covered in like a mucousy substance. And any particles that are suspended in the water stick to the mucus. Then there are cilia that beat and move that mucus over the gill surface to the mouth. Where's the mouth? Over there. And then it's swallowed and it goes through the digestive system, of course. And there's the anus, conveniently placed by the ex-current siphon, right? So that waste material just gets ejected with the water that's forced out. We'll see clams in lab. There, scallops. See right in the centre, there's the adductor muscle that you like to eat. Now you can eat the whole thing. All right, so these are blood clams. So let's have a look at this. What part of the mollusk would you say this is? It's what the clam uses to burrow itself or bury itself. The foot, good, yeah. And it's kind of chewy when you eat a clam, that's the chewy part. All right, and over there you can see the gills. So, blood clams, find them in Shanghai in China. If they're not cooked properly, they're a huge source of hepatitis A and E, typhoid and dysentery. In the list of top 10 food killers in the world, they're in the top 10. I can't remember where they are, but they're in the top 10 causes of death from food, blood clams. And here's number 10, McDonald's. All right, that's right. The number one killer in the States isn't drunk driving or murder. It's the result of fast food, high cholesterol, diabetes, heart disease, hyperobesity. In the end, most of it comes back to those 3 times a day cravings for cheap burgers and fries. As deadly as Tetrodotoxin, which is produced by a blue-ringed octopus, or cyanide. It just works more slowly.
Actually, it depends on your age. Below about 25, it is car wrecks.
What is?
The number one cause of death.
Car accidents?
Mm-hmm.
All right. I guess
Most in the
overall, though,
US.
the number one, well,
The number one-
50% of deaths
Yeah, the number one overall is heart failure.
cardiovascular.
But below the age of 25, it shifts.
Okay. All right. So blood clams are in 1 through 9. All right. So how many of you popped out between lab and grabbed a burger and fries? All right. Ugh, it's killing you slowly. All right. I think this video is worth watching. So this is a scallop, and you can see these little sensory spots. They're the ones that respond to difference in light intensity. But it's quite amazing how they're able to escape. So there's a clam. Scallop, sorry. Off it goes. gets better. Didn't even see that one, eh? That buried one. All right. So that's their escape mechanism. All right, so it's the mantle cavity then where you find the gills. They're the gas exchange organ, but in the bivalves, they're also used for feeding. So I want to take a little look at zebra mussels in Arizona. All right, and that is the right way to say it. Zebra, by the way, not zebra, just so you know. All right. Now, zebra mussels are native to the Ukraine, and they've spread very quickly worldwide in a very short period of time. They were first introduced into the US in 1988, and they are a huge problem. I believe they made their way into the Great Lakes in boats. And once they made their way into the Great Lakes waterways, and they rapidly spread. All right? And they're a huge problem. So they're filter feeders, all right? But they attach to everything they can possibly attach to, and they breed prolifically. And they apparently don't have a lot of predators or diseases because they're an introduced species. Now, has anybody ever been to the beach and collected mussels, for example? Anybody collected mussels on the beach? No? Yeah. So you've got to tug them quite hard, haven't you? They use these proteins, which are these fibers, to anchor themselves to a rock, and they're very effective at doing it. Right? They've got to survive the intertidal, and they've got to survive storms. And zebra mussels are not too dissimilar. They use these fibers to anchor themselves to a substrate, and they're strong. They don't break off very easily. They get their name, obviously, because that kind of zebra striping. So this was a chair plucked out of one of the lakes in Arizona, just covered in these zebra mussels. And so many of our lakes are used for flood control, and many of them are used for reservoirs for drinking water. And as a result, all of them have channels that the water's got to flow down. Some of our lakes we use to get hydroelectric power from. And again, the water always has to flow through gates and channels somehow. And they get absolutely clogged and blocked with these zebra mussels. They're a huge problem. All right.
So what in the Ukraine is the standard predator of the zebra?
I don't know. I really don't. So very often when you've got an organism in its natural environment and it's not a problem, it's at low numbers, it's usually because of competition from perhaps feeding. So maybe there's a competitive mollusk that's maybe not such a problem, or there's some kind of disease or a predator that keeps it in check. And I don't know what it is. So you think the obvious solution might be, well, let's go to the Ukraine, see what feeds on them, and then bring them over here, right?
That's not a good idea.
So biocontrol like that sometimes works really well. There's a good example in Australia of a cactus, and a punchy cactus that went out of control, and they introduced a moth, and the larvae ate the cactus. And it was a very good biocontrol. The myxomatosis virus in bunny rabbits, it was kind of effective, but then the bunny rabbits developed immunity. And then their biocontrol was completely flopped. So what was it? In Hawaii, I think it was rats, and they brought in a biocontrol. I can't remember the full story, but anyway, one of them was nocturnal, one of them was diurnal. And so the one that they wanted to eat the other, well, one was active in the daytime. I think it was, I don't know.
Monkeys.
Was it mongoose? Is that what it was?
Yeah.
They brought in mongoose, which are diurnal. Rats are nocturnal. They thought the mongoose would kill the rats, but of course they're active at different times of the day, so it didn't quite work. And I think the mongoose ended up being a huge problem, didn't they? Because they ate the eggs of ground-dwelling birds. And many of those birds hadn't evolved to build their nests up in trees because there were no ground-dwelling predators. In a lot of the Hawaiian Islands, there were no snakes naturally. We introduced snakes. So we might introduce the snakes to take care of the rats because the mongoose wouldn't. But then that was a problem because they ate birds and the birds ate
Is
on the
that
ground.
the plural of mongoose?
Mongoose? I don't know. Might be. It sounds better than mongooses. Right? All right. So, of course, we get pearls from bivalve mollusks. All right. We get pearls from certain kinds of oysters. And what you can do is the way they make pearls, they get a small piece of grit and they put it between the shell and the mantle. And what the animal does is it sort of sees that as an irritant on the mantle, and then it coats it in the same kind of substance that it uses to coat the mantle. And so it just builds up layer upon layer upon layer, and you get that beautiful pearl. Yep.
So how long does it take for that to happen?
I don't know. I really don't. I imagine it's got to depend on conditions and the species. I can't imagine that long because we've got artificial pearls. You know, we keep these bivalves in tanks and we culture these pearls. So I can't imagine they want to wait that
Cultured
long.
pearls and artificial pearls aren't the same thing.
Yeah. All right. So, class Cephalopoda, the Cephalopods. Cephalopod literally means head, foot. And these are carnivores. So the gastropods, primarily herbivorous, although not all of them, like the cone snails. The bivalves, primarily suspension feeders, but these guys are carnivores. And they've got beak-like jaws. And when we do our squid dissection, we're going to pull out the beak, and it's quite delicate on the little squids, but on the bigger mollusks, it can be quite tough. And they've got tentacles. Usually the tentacles have got suckers. Tentacles are very muscular, very strong, and the suckers are also used to grab and hold on to prey. And it's the tentacles and the suckers around the mouth which are the modified foot.
Technically, only squids have tentacles, and all the others just have arms. Or sorry, yeah, squids and the spirally ones.
Nautilus.
Nautilus, thank you.
So then,
Yes,
so the arms of an octopus, they don't have.
They're arms. They're just not. They're not called tentacles.
Cuttlefish have also got tentacles, though, right?
Yes, they do. I think. Maybe.
All right. So the saliva of many of these then contains poisons. Okay.
Actually, they contain venoms. Poisons are ingested.
So a venom is a toxin that's injected,
Yes.
right? So these guys have got a saliva. So when they bite, it's not really injected.
But it's not ingested either.
It's not ingested.
It's
So
a poison.
I don't know if it would be a venom, honestly. I don't. I still think it's a poison. Don't. It's a good technical distinction to make between a venom and a poison. All right, a venom is injected, but this is in their saliva, so when they bite you, I don't think all poisons need to be ingested. You can absorb poisons through the skin. I'm still going to go with a poison because it's not actually injected. There's nothing to inject it. But what we do know is that some of the poisons in these salivas are produced by symbiotic bacteria. All right. So the story's not been fully worked out, but the blue-ringed octopus, they believe there are bacteria that are producing the toxins or these poisons in their saliva. Okay, so it's the modified foot that forms a siphon, and in lab we'll see the siphon, and the siphon is primarily used for locomotion. It's a way that it can eject water from the mantle and propel itself. Some of them have got a shell, but it's either internal or it's gone, it's lost. So an octopus doesn't really have an internal shell, but a cuttlefish has quite a big internal shell, and a squid has a much smaller, finer internal shell that we'll look at and see. So it's the only mollusk to have a closed circulatory system, and that means that the blood is entirely enclosed within vessels, doesn't come in direct contact with the body tissues, as in an open circulatory system. That means there are vessels, and usually to get the blood around the vessels, there's usually a pump, and that pump we will usually call a heart. And they've got a very well-developed brain and nervous system. In fact, an awful lot of the early research on neurons and how neurons worked was done on the giant axons of squid. They've got amazing eyes. When we do our squid dissection, you'll see the eyes, they're huge relative to the size of the animal. And when you cut into the eye, you can actually pull out the lens, but they've got remarkably good vision. All right, so here's our diagram of our cephalopod then. So this is mostly the modified foot. It's a squid. There's the eye. Let's see. So there's the mouth. I know they've labelled a radula on this diagram. So, but I'm not quite. I guess I'll go with it. But they usually have jaws, and it's beak-like jaws. Not really a scraping thing, but they can move them like this, and they usually just break off parts of whatever it is they fed on. And then they'll swallow it, of course. And there's the anloplin right by the siphon. So water comes into the mantle cavity, it circulates, you've got gills where gas exchange occurs, and then it's pushed out of the siphon, right, together with wastes. Okay. All right, so there's the blue-ringed octopus. How big do you think these guys are? They're tiny. They're like golf ball size. All right, and they have a crazy reputation for what?
Being extremely venomous.
Is it venom or poison? I don't know. We'll have to figure that one out, won't we? But their saliva is very, very toxic. All right, and it is, and it contains something called a tetrodotoxin. And when we look at the chemical structure of the tetrodotoxin, it's chemically similar to substances that certain bacteria produce. And it was that was clues to make us think, well, maybe it's not the octopus producing it. Maybe there are symbiotic bacteria there that are actually producing it for them, or producing something that the octopus can then modify. But they are very tiny. Then we've got squid. I think it's a Humboldt squid. And you can very clearly see the siphon. So water goes in there, and it's ejected out there. And there we've got squid. Calamari. When you eat calamari, you're eating squid. You're actually eating the mantle, right? The mantle forms like a tube, and you can cut it to form those rings. Delicious.
Ew.
Who said ew? Not into
I'm
it?
vegan.
All right, okay, all right. All right. Again, I'm not even going to try with any of these because the BBC moved all their stuff. But you can look them up if you want. All right, so these are the cephalopods then. Oh, you have to look at, it's one of these cuttlefish ones.
Is it a TED Talk one?
It might be on a TED Talk. I don't know though. It's David Attenborough narrating it, and it's about cuttlefish mating. And you've got one that mimics a female, all right? So the big giant male thinks that he's got a female and another female coming in. But when that little male mimics a female, he gets in there and sneakily mates with the real female and then gets away. Yeah, it's cool. All right, so I do want to show you this, and we'll round out with this one since it's a nice little clip. There it is, the infamous blue-ringed octopus. Maybe not. So they're tiny. All right, we'll wrap it up there. So don't run away. I need to give you back your labs. All right?